Afternoon. Well, first of all, I would like to thank Norbert for the invitation. It's a great honor to be here and to learn from Professors Merab and Patrick. And I got very impressed with the presentation and the data. Today I'm going to talk about optogenetic investigation of gamma oscillations in the medial prefrontal cortex of urethane anesthetized mice. This is I collect data from my internship abroad. I did at the Marie Carlen's lab in Karolinska Institute, Sweden. I started my PhD in 2010. And then I got a fellowship to do this internship in 2013. And then I came back to Brazil in 2014, and then I could finish my PhD. So this is me almost freezing during the winter, and this is really beautiful sunset. Actually, that was 9 p.m. in Stockholm. That's really, really wonderful landscape. And this is the Department of Neuroscience in Karolinska. Actually, the summer in Stockholm is pretty much similar the weather with the winter here in Brazil, right? Okay, this I think you guys already know. So. In the brain, the mammalian brain has billions of neurons, and you can find thousands of subtypes of cells. For example, these cells are basket cells, and these cells could be pyramidal cells, for example, excitatory cells. And you can target specifically subtypes of cells with optogenetics. So, for example, you shine light in the whole population of cells, but you can activate, for example, just the basket cells. So this is precision of spatial. Actually, this is spatial precision, but also we can find or give temporal precision. So every time that you shine light in the population, you can get one spike, for example, one actual potential. So for example, you shine light and you activate the cells. And this is actually very important because you can probe mechanisms about the genesis of some oscillations in the brain. And you cannot do that if you apply electrical stimulation, for example. So one thing that is important to know is about the hypothesis of NMDA receptor hypofunction on GABAergic neurons for schizophrenia. So there are studies that in human subjects supporting the idea that there is an NMDA hypofunction in schizophrenia. So it's more related to dysfunction than the number of the receptors. For example, in some imbalance between some subunits of NMDA receptors. Also, there is some recent evidence supporting the role of PV neurons in gamma oscillations production. And I will talk also about some circuit models of inhibition based on gamma oscillations. And there are distinct properties of AMPA receptors and NMDA receptors mediate synaptic response and their contributions to excitation of PV neurons. And finally, there are studies that test the effects of NMDA antagonists on gamma oscillations. So, for example, ketamine. If you treat human subject with ketamine, you can reproduce most of the symptoms in schizophrenia. For example, some positive symptoms, cognitive deficits, negative symptoms. And also, if you inject ketamine in a rat, for example, you can produce some cognitive deaths like working memory deaths and also hyperlocomotion stereotypes. But the problem is that ketamine has binding for many other receptors, for example, opioid receptors, G2 receptors, and this becomes very difficult to probe NMDA-based mechanisms in gamma oscillations. You can also test with MK801, that is a more specific NMDA antagonist, even though you cannot produce temporal precision in activation. So that's why optogenetics is important to probe this problem. So parvalbumin positive cells precisely control the activity of cortical pyramidal cells. This is a nice video from Harry Lab in France. And here you can see parvalbuminergic interneuron firing here. And during this firing, there is a silence of the excitatory neuron. So, and this excitatory neuron only fires when the parvalbuminergic neuron is not active. So, and this here you can see gamma oscillations recorded in local field potential electrode extracellular. And these are pyramidal cells and interneurons, basket cells. And these basket cells, they have also gap junctions, so they display electrical synapses. So they can fire very fast and coupled. And this could be the base of generation of synchronized neuroactivity. And one important thing is but how to test this mechanism? So Cardan et al. They show that light-driven activation of fast spiking cells selectivity amplifies gamma oscillations. So this is the first experiment they performed with PV-Cree mice. So they transfected only PV cells with channel rhodopsin, and they apply different frequencies of stimulations from 8 to 40 hertz. And also they tested CAN kinase 2 Cree mice to drive specifically excitatory cells. This was in the somatosensory cortex. Okay. And here you can see if you shine light at 8 hertz stimulation in PV-Cree mice, no effect. You can see no effect actually. But if you apply 8 hertz stimulation in CAN kinase Cree, you can amplify this oscillation. On the other hand, if you apply 40 hertz stimulation in PV-Cree mice, you amplify gamma oscillations. And then nothing happens with CAN kinase Cree. So if you plot frequency of stimulation and this y-axis is LFP power ratio, it means post-stimulation divided by the baseline condition, okay? So here is possible to see if you stimulate at around 10 or 8 hertz, you can get 7-fold enhancement of power around 10 hertz, but this is regular spiking stimulation. And then when you go faster with stimulation, with faster frequency of stimulations, you cannot see actually any significant effect. But if you apply in PV-Cree mice, you can see that there is a specific enhancement of gamma oscillations in the range, in the gamma range, okay, from 30 to 60 hertz. But how to understand this data? So one possibility is that the excitatory inputs to the PV neurons could be crucial for the gamma oscillations production. And the other possibility is that only the interactions between the interplay between PV neurons is sufficient to produce gamma oscillations. If that's true, if you selective knockout GABA receptors in PV neurons, you should see some depth in gamma oscillations production, right? And it has been demonstrated that actually if you delete GABA A receptors, there is no effect in gamma oscillations, but in theta oscillations actually. On the other hand, if you actually, there is two evidence. One showing that if you knock out GluR-D receptors, that is important for AMPA receptors in PV neurons specifically, you have very severe decrease in gamma oscillations production. And also, if you knock out NR1 subunit, that's obligatory subunit of NMDA receptors, also you can find deficits in gamma, in the evoked gamma oscillations. So it's important to bear in mind the difference between AMPA and NMDA receptors. And in the resting potential of the membrane, AMPA receptors can be activated by glutamate, but NMDA receptors is blocked with magnesium. So it means that when PV neurons receive glutamate inputs from other neurons, the first receptor that's going to be activated is the AMPA receptors, okay? And then it's going to be NMDA receptors. So maybe for gamma oscillations production, AMPA receptors is more important in PV neurons than NMDA receptors. So this is the evidence showing that actually they show with GluR knockout animals, specifically in PV neurons. This was in vitro experiment. The gamma oscillations was induced by kainate bath. And you can see here crescent concentrations of kainate. And this is the wild type animal. You can see the enhancement of gamma oscillations, and the knockout animal cannot show the same. Also, mice lacking NMDA neurotransmission only in PV cells exhibit enhancement of baseline cortical gamma vertins. So in this case, there is an enhancement, not a decrease in gamma. I told you guys that maybe AMPA receptors in PV neurons is more important than NMDA receptors. And then in this case, if there is a lacking of the NMDA neurotransmission, there is actually an enhancement of gamma oscillations. And you can also produce this by injecting MK801. That's a very specific NMDA antagonist. But the main problem here is that you can produce, you can see enhancement of gamma oscillations at baseline condition. But you can also, if you drive PV cells to produce gamma oscillations like I showed before. And this is the PV Cree animals. So from 30 to 60, you can see the enhancement of power. But the PV Cree NR1 knockout show a depth in the evoked gamma oscillations by optogenetics. So you can see both. You can see baseline enhancement of gamma and decrease in optogenetic evoked gamma oscillations. And also, if you apply MK801 here, IP, you can see actually an enhancement of gamma oscillations in NR1 flux, that's animal control, like PV3. And then actually a decrease in gamma power in PV3 and N1. So there is a reduced sensitivity to the effects of NMDAR antagonists on gamma oscillations, which means that the target of the MK01 should be NMDA receptors at PV3 neurons. So actually, if you treat some health person with ketamine, you will see actually enhancement of gamma oscillations. But also in schizophrenia, in patients with schizophrenia, it has been reported that there is a reduction in the gamma evoked oscillations by, this was an auditory forced choice discrimination task. That in control animals you can see actually a very robust enhancement of gamma oscillations by the auditory stimulus, but in schizophrenia patients there is a decrease in this evoked. So this could be compared with the data from optogenetics induction of gamma oscillations and the decrease of these gamma oscillations in PV3 and N1. So, and also, but one problem with this is that both Cardin and Carlin, they has been shown this data in the primary somatosensory barrier cortex. And the prefrontal cortex is more related to the schizophrenia condition, right? So some open questions is it be possible to evoke gamma oscillations in prefrontal cortex circuits by driving PV cells? And if yes, how important is the NMDAR retransmission in the prefrontal cortical PV cells for gamma oscillations expression? That was my very basic two questions that I used to guide my training at Mari's lab. So we aim to test for the possibility of gamma oscillations enhancement by driving PV cells in the medial prefrontal cortex of urethane anesthetized mice. And also we aim to investigate the role of NMDA receptors specifically in PV neurons during basal and optogenetically evoked gamma oscillations. So for this, it's important to mention that I was doing a training in optogenetics. So I was, at that time, I used to know nothing about optogenetics. So I had to follow these steps. So first, it's important to select the correct opsin. So and we chose channel rhodopsin. This is not actually the best alternative, but it was the first we had to do. And then we chose our target cell type was parvalbumin positive cells in PV neurons. So to do that, you just need to inject. First, you need a transgenic mice pre-expressing your cell of interest, for example, PV3 animal. And then you do the virus injection in the area of interest. So you inject the virus in the prefrontal cortex of PV3 mice. So you are going to get parvalbumin positive cells and specifically expression of channel rhodopsin in these cells. Okay. And then you have to be careful in choosing the protocol of how to derive the light. So we chose blue light. The power is important. And the diameter of the optical fiber also. And this, we set the temporal parameters. This is from 8 to 90 Hertz stimulation. And then you perform the plane validation. And the plane validation is important to do microscopy with the immunohistochemistry to looking for specificity of the transfection. And also do electrophysiology to confirm the optogenetic manipulation of PV cells. And here, I don't know if it's possible to see, but this is a coronal slice of the prefrontal cortex. Okay, from channel rhodopsin inject animals. The fluorescent marker here is the M cherry, so you can see red here. And the target was the prelimbic area in the prefrontal cortex. But when we remove the needle, I think some virus leaks here, right? So you can see transfection in this whole path. And these are two examples of the control virus injected. The control virus is the virus that has the promoter, but doesn't include channel rhodopsin, just the promoter and the fluorescent reporter. Okay. And we also used the CRELOX-P strategy to transfect specifically PV neurons. And here you can see the confirmation of the transfection. We did some PV staining and check for overlap. And here our setup, we used silicon probes in the prefrontal cortex, and then another arm to hold the fiber. And the animal was anesthetized first under isoflurane, and then we inject urethane slowly, and then we only replace isoflurane by urethane. And then we keep the animal above a heat pad to keep the temperature constant. Our data, we used silicon probes 4 chunks, 2 tetrodes per chunk, so this makes 32 channels, right? We put the silicon probes in the prefrontal cortex and the fiber in an angle. And here you can see the software of the neural links and how we sorted the units. So you can actually sort lots of units using these silicon probes. Average of 20 units, 30 units is really good actually. So here you can see some traces from the LFP recorded in the prefrontal cortex. And the urethane, the animals display these slow wave patterns. And you can see also the 25 cells I got from one animal. And it's really nice to see that these cells is very coupled with the up phase of the wave. And then if you do a correlation between spikes and phase of the wave, you can see that there is actually a perfect correlation around 0. There's almost no delay. And you can plot the lag here and compare, for example, PV3 and NR1 PV3. We got 54 cells in NR1 PV3 and 27 cells in PV3 animals. And also we perform coherence between spikes and local field potential. So far we didn't see any difference between PV3 and NR1 PV3 animals. But we actually did see some theta enhancement in the NR1 PV3 animals, and a trend towards enhancement of beta and low gamma oscillations. This is 5 animals in one group and 3 animals in another group. This is a work in progress. I'm still doing the data analysis. Sorry. Sorry. Okay. So this is an example of one fast spike spiking cell I got. So as I use a tetrode orientation, you can actually see if you can sort the units by comparing the amplitude and the energy of the wave and the waveform shape. Also, you can perform PCA comparisons. So this is one fast spike cell, putative fast spike cell, and we perform many trials with different frequencies of stimulations during 3 seconds. And then sometimes we apply, for example, 8 Hertz or even lower frequency or higher frequency, okay? So here you can see that the cell is actually following almost all the time the stimulation protocol. But with the higher frequency, there is some enhancement of the activity, and then it's not possible to follow all the pills you apply. Sometimes you lose the spike because the frequency is too high. And then also I could sort the cells that is putative fast spikes by the waveform shape and regular spike cells. And this is important to do because, for example, you can see here this blue one that is a putative fast spike is the one that I got that was activated by light, so should be fast spike. And these orange ones, that's putative regular spike, was inhibited by light. So this should be pyramidal cells that receive projections from fast spike cells. And this is actually easier to find examples like that was inhibited than fast spike cells that is responding to the light. Because it is difficult to find fast spike cells, and fast spike cells that respond to light is even harder, right? So you can see some examples here. This is regular spike cells that is being inhibited by 3 seconds stimulation. And this is one example of fast spike cells that was driven by light. And next, finally, we try to respond to our first aim, that is it possible to induce gamma oscillations by driving PV cells in the prefrontal cortex? It seems that it is possible. So you can see here, this is the animals that were injected with control virus. So no channel rhodopsin, so you apply light, 8 Hz, 40 Hz, doesn't matter, you can't see any effect. But with the channel rhodopsin injected animals, there is a robust enhancement of 40 Hz oscillations when you apply 40 Hz. So you can see here power spectral density around 40 Hz. So photostimulation for activation of channel rhodopsin expressing PV neurons imposes transitory decrease in the firing rate of nearby regular spiking, putative pyramidal neurons, and causes a specific enhancement of gamma band power in the medial prefrontal cortex. So in order to test for the importance of NMDA transmission in the prefrontal cortical PV cells in the evoked gamma oscillations, PV-CRE and NRM PV-CRE mice will be compared regarding oscillatory patterns and firing rate in the prefrontal cortex. These data analyses are ongoing. So this is a blog that my wife and we wrote to share experience about living in Stockholm. That was a wonderful experience. So we think that's important for the younger students to know a little bit how to get the visa for Sweden, how to the curiosities about food and people, the landscapes, place to visit. So go there and take a look. So this is some pictures. This is the lunching room. Very beautiful. The department, water maze. I performed some experiments there as well. The department. This is from near to my home during, I think it was April. Bus stop. This is actually this is the building I was living. And I think it was like around February, a lot of snow. It's from the window of my place. There's some guys like skating with baby. That was really interesting to see. And some barbecue, you can buy this in the supermarket. This was curious for me. And just some pictures, and we had big celebrations during the summer. Like we put maybe 20 people inside of a very small apartment. It was really nice. Then we played some football. And then I would like to thank you, Professor Marie Carlin. for the support, for receiving me, for the opportunity. And also especially these two guys, Calvin and Hosiok, was the two postdocs that co-supervised me. They could teach me how to do virus injection, how to prepare the fiber, how to do the spike sorting, and all very difficult things that I could learn. This is one of my best friends now. His name is Nicolas. He's now a PhD student. He's from France, but he used to live in Portugal during 10 years, so he speaks Portuguese. So when he arrived, we performed lots of experiments together. And then thanks to him, we are still in collaboration. He's performing experiments there, and I'm doing data analysis here, so we are still in collaboration. And she's Deborah. This is Deborah, is a Brazilian. She helps me a lot as well. We spend the Christmas at her apartment there. Okay, thank you to FAPESP, University of São Paulo and Karolinska, Professor João, Rodrigo, Marie Carlene, Calvin, Hosiok, Deborah, and Nicolas. Thank you for your attention.
Hello. Thanks very much, Clayton. I think I need to come here to start the open discussion. So thanks all. Merab, Patrick, Clayton. In the middle. So the discussion is open for questions, comments, please.
So Ed Boyden's lab and Carl Dijsselrath's lab have both developed red-shifted variants of a variety of these pumps. And those red-shifted variants, at least in the mouse, through a small thinning of the skull window, you can basically activate the entire brain because of the wavelength and the absorption rate. So there are variants of generodopsin that are red-shifted. There are variants of inhibitory ones that are red-shifted that allow exactly that, the non-invasive the less invasive activation or inhibition of cells. And both of those labs have been amazing about making their reagents very accessible and freely available. So that's a big help.
Okay, go ahead.
When you use that approach in which you inject a virus in a region and you simulate with the life in another region, what can you make to avoid possible retrograde action or light simulation to the whole neuron?
Yeah, so the approach that we've done is the opposite. We've inhibited terminals in one region. So there we don't have to worry about backpropagation of action potentials. You're right, that's a big concern for terminal activation. One of the ways that people have proposed to get around that is actually using the DREAD approach, the chemogenetic approach, because you can microinject clozapine oxide at the site of terminals, and that actually does not trigger action potential backpropagation. You get local transmitter release, but not backpropagation. So that's the kind of one of the benefits of the chemogenetic approach. But directly into the brain exactly. So you put a cannula in, and just like you would for a traditional microinjection experiment, you can inject CNO at the site of terminals, and that's one advantage. I don't know any other way to get around it, though. That's a problem of the electrical properties of neurons.
Just to comment on that, I think in some cases first of all, you should know what you are doing actually, and this is exactly what you have to think about when you do this optogenetic studies. But the other thing is that it could be in some cases disadvantage because you don't want the somatic activation, antidromic, but in some cases it's opposite. So you actually want to activate antidromically. I think we don't know yet, but in the data I showed you with this sort of kindling phenomena, we we are not activating dentate with our fibers, we think, because they are located in CA3 area, but it could be that there is this antidromic activation of the dentate, and you have to involve the circuit and also activate dentate, and then it has to get bilateral and so on. So that could be one of the explanations. So, but I think it's important to think about these details. Yeah.
Yeah, there's also an approach developed by Dijssel's group called intersect, and this approach is making use of both Cree and flip recombinases. So what you can do is, imagine that you have kind of two projection regions. Region A projects to both B and C. Depending on where you have flip recombinase and where you have Cree recombinase, you can select for cells that only project to region A, or only project to region B, or project to both region A and region B. And they accomplish this with kind of those same inverted open reading frame constructs where the channel rhodopsin is flipped, but now it's actually controlled by the two recombinases. So depending on the orientation, you'll either get expression or not. So that's another way if you want to target just one projection pathway, if there is a specific projection pathway.
Additional question or comment? Let me ask Merab on the kindling protocol. But for those who are not familiar with the kindling, will you repeat simulation, subthreshold simulation? In the beginning, you don't get any seizure, either behavioral or on the EEG, but suddenly you start getting the so-called after discharge. It's a small amount of activation through the EEG, and with time, the same simulation repeated, it will produce an enhancement or an increase of the length of the after discharge. So you do compare the electrical simulation with the optogenetic simulation. Do you have any behavioral alteration? Is it compatible? Thinking of, for example, Racine scale, which is a seizure severity scale.
Yeah, we haven't done that. I mean, the data I showed you, it was in anesthetized animals that are in the frame. So that would be interesting to look at. But I think what we are looking at is a much more smaller scale compared to what you do with electrical stimulation. Actually, you have much stronger stimulation with electrical kindling compared to here. And our goal was to try to identify this minimal circuit that you have to tap with to induce this type of plasticity that would lead to enhancement of excitability and change in the network. So that is different, and it will be interesting to see what happens in the behavioral level there.
Because anyway, if you compare these kindling protocols, the autogenic kindling is another protocol in which you hit the sound several times, so you repeat the simulation. That's a sensory kindling. The other is electrical kindling. But the comparison usually is with the status epilepticus models, which is a stronger situation, 1 hour and a half, 2 hours of continuous seizures. So in those cases, it will be interesting to block also, to try to block maybe through those pathways, through optogenetics, because you're looking at a circuit level or a network level instead of just a single spot. So how do you comment on that? At the same time, you need to work with the specific places in time course. You have generalization that is driven by connections all the way through the brain. It's going to some other places so far away from the original focal area.
I think you're right. These are two completely different models, and kindling model has been very popular since 70s through 80s. But then the relevance or the clinical relevance of this model has been questioned, because actually you don't get spontaneous seizures unless you kindle 300 times or things like that. So there is a paradigm shift, as I see it, in the epilepsy field, that people tend to use more chronic models, like post-status epilepticus, spontaneous seizures. That doesn't mean that I think for looking at the mechanisms of ictogenesis, for example, acute ictogenesis, these models are still quite useful. And I think depending on which model one uses, one can address different sets of questions. And I think from the clinical perspective, it could be more interesting to go for chronic spontaneous seizures, but that imposes another level of complexity to the experiment, because you don't know when the seizures will arise, and you have to have this kind of closed loop system to address these questions, which is possible, and people are doing it. So, but for us, what was interesting is to see what is the initial sort of minimal circuitry that you can activate, and that would lead to those plastic changes that eventually become permanent. And with the same stimulation, now you have very advanced seizures, so to speak. So this process of, let's call it epileptogenesis, in terms of circuit modulation, was addressed in this type of setup. But I think if you are looking for further and try to target these changes in more chronic model, that would be another level of approaching this.
I would say that there's an order of operation to experiments, and when you're employing a new and more complicated technique, I think it makes sense to start with the model where you have the most experimental control. But I think both of you are absolutely right that it's essential to transition to models that eventually better approximate the human disease condition. I would say that when you look at models like, I have my own slight bias against the status epilepticus models, because it is not all that common that you have a human that undergoes seizures for 2 hours as their first unprovoked seizure. So, excuse me, from the perspective of etiology, it isn't the most relevant model. From the perspective of the amount of damage that it causes with prolonged status, it's not the most relevant model. And actually, when you're looking at neural circuitry, if you want to optogenetically control some cells or microinject drugs into a brain region, if that region is Swiss cheese because the animal has had status for 3 hours, there might not be anything left to... So again, I think for epilepsy in particular, you really need a combination of models that allow you to address the same question from multiple different angles, because any one model is probably going to lead you astray.
John, you have a comment? And including you in the conversation.
Well, actually, unfortunately, I didn't have a chance to attend all the lectures, but from the perspective of physician, I think this debate is extremely important, and I think that I know it's very difficult to work on real pathological conditions. Maybe we're going to have difficult conditions to control, but that's what we have in the clinic. So we cannot, in fact, most of the symptomatic epilepsy cases that we underwent surgery, they have somewhat damage, and we have probably a blood circuit that is pathological. We have cell loss. And I know that optogenetics now, you can control in a very sophisticated manner to understand what is the epileptogenic process. So this is going to help anyway. But to be in a condition to use closed-loop systems, probably we're going to have to also use some models that would be, or at least would have some damage to somewhat replicate what we have in human condition. But I think that all the questions that you asked are very important. We have to go to some less invasive protocols. And one question that I've seen in several can we be sure that with more experience we could, at least in some very controlled situations, use this viral infection in human condition? Or do you think that we are far from that ideal condition?
Thank you very much for this comment. I think I was trying to say basically the same thing, that it is extremely important to know what is clinically relevant and not. And I think Field now has realized that and moving towards closing the gap between clinicians and preclinical researchers. And one of the attempts we have made in Lund is to create Epilepsy Center, where we have this virtual center, so to speak, where we meet with the clinicians, and together with them we define what is the problem and how to deal with it, and how to go and translate then the basic studies into clinic. And I think you're absolutely right. We have to deal with the human situation where there is a damage, where there is chronic progressive epilepsy, and it's pharmacoresistant and so on. Just a little side comment is that we should perhaps use more the tissue from the surgeries as our validation point, because even if we now shift into the epileptic animals, it's not the same as human epileptic tissue. And I think we have very unique opportunity in epilepsy field to really use this tissue to validate our preclinical data. When it comes to the use of viral vector-based gene therapy in general, particularly it has been, for example, AAV viral vectors have been used in Parkinson's disease with a safe outcome, and there are thoughts of doing it. For example, also in Lund we have this idea that perhaps we could do this gene therapy approach, not necessarily with optogenetics in the first place. As I see optogenetics at this point is that you may want to delineate more the mechanisms and identify your targets more precisely, and then go with the other techniques, either with pharmacokinetics or pharmacochemistry, like Patrick was presenting, for example, or some other means, or even with optogenetics, but in the future. But when it comes to the gene therapy, I think we are quite close to clinical trials, and not necessarily, as I said, with optogenetics, but with the vectors that produce sort of endogenous factors like neuropeptides, for example, or some others that are native for the brain, and probably you have much less chances of getting immune responses. Even cholinergic receptors that are modulated, they are probably less risky compared to opsins. So that particular concern has to be really taken seriously. But on the other hand, that shouldn't preclude us going into clinic and really trying it, and particularly perhaps using this first step of validation with patients that we take tissue out, so one can test this tissue. Now we show that it's possible to do it in a long run, like for several weeks, where you can express your viral vectors and test the effects. But also identifying the patient group that provides this type of safety. And I think those that are planned for the surgery gives, again, a unique possibility for testing this in a more safe way, because then you can always still take out this tissue. And the viral vectors, they're not spreading really that much. For example, AAV, it is quite local. So as soon as you have tested the safety and perhaps even efficacy, okay, one can discuss whether if it's very efficacious, then one shouldn't take it out, but you can always take it out anyway. And I think that would be the first group of the patients to test this viral approach, because you have this additional safety aspect. And that gives you another possibility to really look then at the tissue and see what happens. How was the expression? Was there any problem? Did you affect the tissue, induce more inflammation, for example, or things like that? But it will be very difficult to compete with the surgery as a treatment per se, because it's very effective. I don't know if you would agree, but most surgeons would say that it is. And so maybe that's not the primary target. Once you have defined that it's safe and it's efficacious, perhaps the primary target is those patients that cannot be operated. I don't know that, but I mean...
But maybe it's like, I can imagine in 50 years, probably we have like a whole group of scientists that see, can you imagine that in the turn of the century, people still do surgery to remove a whole chunk of the temporal lobe to treat dyslexia? So if we cannot at least challenge our imagination to see that it's still a really rude, crude procedure. So far, we are taking out the anterior part of the hippocampus and the temporal lobe and the body. It's a huge chunk.
It's an amygdala problem.
Yeah. Amygdala together. So it's not
I couldn't agree more, and this is what I think, that in my mind any kind of treatment is better than just taking a tissue and throw it away. But we have to face the reality that the treatment you are proposing and something that is coming in should compete to existing treatment and should be at least as efficacious as the one, or at least should be less invasive, or should be more benefit. Risk and benefit ratio should be higher, or it has to be some way better to really get established as a treatment. Experimentally, you can do, of course, things, but to be established as a treatment, it has to compete with existing treatment and be as efficacious as the existing treatment. But I couldn't agree more than what you are saying.
But anyway, I think one of the things, we have tons of tissue that have not been analyzed, either by morphology, either by optogenetics, or by electrophysiology. So still we have, without not doing any additional surgery, you have tons of tissue all over the world that were not analyzed. But probably the future will be something different. It's non-invasive, with computational tools, spike detection, closed loop, etc.
So I think there's one other thing to think about in the context of temporal lobectomy or selective hippocampomygdalectomy, and versus the optogenetics, which is, it's still not entirely clear that the tissue is something that you want to have around, not necessarily for the purposes of epilepsy, but for the purposes of cognition. And the group at the Montreal, including Mary Pat McAndrews, have spent a lot of time looking at cognitive predictors of whether or not a patient will benefit cognitively from temporal lobe resection. So I think we still need to know more, not just about the seizure side, but also about the effect of damaged tissue on memory and other cognitive domains to know whether or not something like the optogenetic intervention or another intervention may actually be superior, because it may not actually in the end. You may not want to stimulate. You may want to remove the bad tissue. And until we have more data on that, I think that's another kind of limiting.
When I was discussing at Mari's lab about how to choose the best parameter of stimulation, for example, frequency, duration of the pulse, power of the light, we found difficult to to see, we found actually very controversial information in the literature about the parameters of the stimulation. I would like to hear from Merab and Patrick about how to, maybe we should talk more about or share experience about the parameters, because for example, for gamma oscillations induction, we still really don't know if that was the best option to do. I saw some papers from Bouchak lab. He's applying sine wave stimulation. That seems to be much better for me than the square shaped pills. And then today I saw you perform stimulation, Patrick. You perform it with 100 hertz stimulation. So could you comment a little bit about how to choose the parameters if you had this problem as well?
Yeah, we don't know that. That's the answer. And we're in our particular experiment, to be very concrete, we tried from 10 hertz up to 100 hertz, and basically we saw the similar effect when it comes to inhibiting epileptiform activity in this particular situation. So, but I've heard there are some data showing that lower frequency stimulations in this model could in fact trigger in PV animals could trigger epileptiform activity. The hint of that was also in our data, showing that this first switch on of the light could actually trigger action potentials and so on. So one has to be very explorative, I think, and use not only different frequencies but also different intensities to avoid, for example, this first, if you want to inhibit epileptiform activity, to avoid this first very synchronous release by ramping the light, so changing the intensity, playing around with the frequency. I think we still are in the beginning of this, and we need to do more explorative work, and sharing the information would be also very helpful and one way to go.
I would say that the way we've addressed it is much the same. We've tried a variety of stimulation frequencies, for the activation, as low as 1 hertz, as high as 100 hertz. There are complications associated with the higher frequency stimulation. Depending on the channel-rhodopsin variant, you can have a decreasing fidelity of spike, driving spikes. So you can wind up not getting very faithful spike representation. So if that's something that matters to you, you need to think about which variant you're using. And something like the Kronos variant from Ed Boyden's lab has very, very faithful single spike resolution, even up at those high frequencies. I would say that the other thing is, on the inhibitory side, again, it was kind of exploring. What we found is when we dropped our light delivery frequency below 100 hertz in the substantia nigra, we no longer silenced the cells. And I think it depends on the population that you're dealing with. So the neurons in the nigra are tonically active. They have a high basal firing rate. They have strong driving forces that keep them tonically active. They only pause physiologically when you have a huge barrage of GABA coming from the striatum. So when we dropped our frequencies lower than that, we couldn't shut them off. So that's why we stuck with 100 hertz for that, because anything less didn't reliably silence them.
Have you tried longer pills?
The pulse, in terms of pulse
Like, for example, 3 seconds pills.
Yeah, so we've also done continuous stimulation where we've turned the light on and just left it on for the entire experiment. The other way of dealing with that are the step function options. I don't know, Mirab, if you use them?
Yeah, I just wanted to comment on that.
Yeah.
So there are these SSFOs, the step options. You probably know about this. So these ones are not inactivated spontaneously, but rather require another light pulse. So when you pulse them with certain light spectrum, they get activated and stay activated without any light. And then to deactivate them, you have to shine them again with a different spectrum. So that's another thing one could perhaps explore and see how that will work. And I think there are now possibilities both for depolarizing ones and hyperpolarizing ones. I think this was mentioned, it's not published yet, but they have this new options coming out with a step function, which are channel options with a chloride permeability.
I can't wait for it because it solves a lot of problems in terms of long-term tissue heating.
Yeah. So that could be another thing to test in more systematic way, I think.
I have a question for the two of you.
This is a small comment on the types of simulation or parameters. I mean, when you talk about sine wave, I remember in electrical simulation, that's true. It's proven that the sine wave is more protective in terms of non-lesion in that area. Usually we use periodic simulation, but non-periodic simulation, such as scramble simulation, is also quite effective. Electrical scramble simulation. So what about optogenetic scramble simulation, non-periodic? I think because our mirror is always the experience made with electrical simulation. It's kind of repeating with them and try to know what are the advantages of using optogenetics without causing the same problems we are already exposed with electrical simulation.
Absolutely.
So I was wondering if either of the two of you have, or how the two of you have dealt with issues with the Becquerel effect or the photovoltaic effect in terms of your recording when you're shining light across an electrode, and how do you deal with the artifact? Because it's something that we struggle with a little bit.
Yeah, so the question was that the photostimulation may induce some artifacts in recording. Well, we were aware of it and we did some control experiments, but in fact we haven't really been troubled by this. And I think it very much depends on the intensity of the light and how close the electrode is. Yeah, I can't comment more than that we haven't really experienced too much of the artifacts. And that probably depends on the intensity of the light and diameter of the fiber, things like that, and how close they are, and what is the distance between the tips and these kind of things.
Yeah, I spent maybe the first 3 or 4 months of my training trying to remove the light artifacts. Okay. So what we did was first trying to put the fiber with a more distance from the silicon probe, and then reduce the power as much as possible, and trying to put the fiber in an angle to avoid the light shine directly to the metal contact, and then we could remove most of the artifacts. But I think for our experience, it's also good to do the stimulation in a specific frequency and looking at another frequency. So this would be better. So trying to induce gamma oscillations by applying 8 hertz stimulation. That we could see that's not possible, but we could also try apply, for example, 60 hertz stimulation and take a look in 35 to 40 hertz stimulation. So we could try to see if we can indeed enhance in gamma oscillations or if we are just seeing light artifact. It was our main concern at the beginning.
Alguma pergunta? Vocês estão caladinhos aí, podiam fazer em português ou em espanhol. A gente faz a tradução aqui. Algum comentário? Sem? Poliana? O colega aqui perguntou no começo. Vai. Ok, vai.
Is there any study trying to modify chemistry in neurons instead of making it permanent, licensing?
So Certainly in cell culture, people have done this with plasmid-based approaches instead of viral-based approaches. I don't know that anybody's done an equivalent approach in vivo. The way that that's been done for other systems using AAV vectors is with a TET-on, TET-off sort of system, or a multi-virus system where you can have CRE under the control of something inducible like TET. So if you, theoretically in humans at some point, if you had a negative effect, you could give a single high dose of tetracycline and excise the channel rhodopsin cassette from the genome. But to the best of my knowledge, nobody's tried that yet with the optogenetic approach. Do you know if anything like that's been done? But I think that would be the likely approach to it because it's been tried in other contexts, not for therapies, but mostly as a scientific tool.
I think it raises an interesting idea that if you want to perhaps have a transient effect on the circuitry, and that may be enough to modulate, and then you don't want the expression to go on. So in that case, I think one could simply use the viruses that do that temporarily, HSV viruses. So they, for example, go up in their expression, and after, I don't know, a few weeks, they decrease their expression. So then you have your window where you could affect the circuitry, and then it will be removed. But I don't think we are at that stage at present. So people haven't really tried that. But people have tried it with other genes, so it's possible. For example, Michele Simurato has tried a transient expression of BDNF and or FGF and something else in combination. I don't remember now, but it was a transient effect during epileptogenesis that prevented chronic phase. So that could be an interesting approach to have a window where you modulate your circuitry and then you stop expression.
I think the caveat with some of the other vectors is just the safety of the vector to the tissue. AAV is pretty well tolerated, at least in kind of the broader experience. Lentiviral vectors, at least in our hands, and I know in several other groups, cause a little bit more damage. And HSV, depending on what brain region it's going into, can also cause a good bit of damage. So I think if you were to go that route and try one of the other vectors, you would have to really pay careful attention to any off-target effects that might be going along with it. And I know that people do these controls for those experiments, but it's important, I think, to remember that switching vectors does not necessarily mean that you're going to have equivalent safety profiles.
Any additional comments, questions? Okay, just 3 questions.
Yes. Yeah, I think Schindler, they have done channel rhodopsin expression in the newborn cells and looked at the efferent synaptic connections to the other cells. And then they found that they do get integrated in terms of efferent synapses as well. So one good way of using optogenetics is to really, and we have used this to see, for example, when transplanted cells, if they are integrated into the circuitry. So you can do, you can transduce the host tissue with channel rhodopsin, for example, and then transplant your stem cells or whatever you're transplanting. And then if you take slices and shine on the slice, then only the host cells will be activated. And you know for sure, if you record from the transplanted cell, that these synaptic connections are coming from the host. So then you have a sort of more or less really hard evidence that there is a synaptic connection. And the other way around has been a little bit more difficult because the stem cells and developing cells don't tolerate very well the opsins. But people have achieved it, and this inducible expression could be one way of going there. But definitely yes, I mean, and it will be extremely interesting to look in different pathological situations, for example, how these cells are integrated, and also perhaps tackle their activation. What would be the consequences of their activation in terms of behavior or pathological situations? So I'm sure there is ongoing work in that direction.
I think the other thing that's been done that's slightly related to that is tagging cells that are active at a particular moment in time. So you have CRE or something under the control of an immediate early gene coupled with an estrogen receptor on-off system or a TET on-off system, so that you can get really precise control of expression of your opsin in a cell that was active during, let's say, a memory formation. This has been done by the group at MIT in a couple of different cases now.
And then you can activate
exactly.
Exactly those cells that have been activated and induce this similar behavior. It's amazing.
Very nice. Welcome. What is Coimbra? Any questions? No. Replay for you. I think it's time to close, maybe. You have any additional comments? What about the single 2 lines statement, a recommendation for the young people, those interested in coming into this area? 2 statements, 20 words, saying your final comment on this gathering.
Should they go to Sweden?
Yeah, sure. You should go to Sweden. It's a wonderful place. It's easy to get the visa, for example. This could be a big problem if you go to UK, for example.
Australia, Canada.
Yeah. And the nice thing is that optogenetics has this idea of the open science. So you can find information in the internet easily. So at Boyd and Dijsselhoff, they are sharing the virus, the vectors, the tools. You can find protocols there. You can find everything. So I think this idea of open science is really, really interesting. And for the younger generation, I think we should stick with this idea of you do not just keep the information for your lab, but try to share, try to put on the internet. And I think this is important for example, if you receive public resource, this is important to try to put the information public as faster as possible, right? That's my idea.
My comment, when it comes to optogenetics, I started it in 2006 or 7, and the first publication came out in 2009. So it takes time to get into the field, and you have struggled with it a little bit less than I have, because you were already in the established lab. I was just establishing it myself. So it's much easier, and I think it's a really powerful tool, but you have to really know what you're doing and think of all caveats or underwater stones that this technique, not different from other techniques. You have to know what you're doing, and it may look very simple when you listen to the presentation, but you have to really know and realize what you're doing. But I think one has to go for it because it's quite powerful. There are new techniques emerging, like you mentioned, DREADS, for example. If you don't want a millisecond precision in time, I think DREADS could be another alternative. So there are plethora of techniques now which are really amazing what you can do with them. And the major thing is to identify your scientific question, of course, and then try to use the technique that is most appropriate. And optogenetics is one of those that is very useful.
I'll echo the fact that setting this up for the first time in your lab is not a trivial experience. We started in 2010, 2011, and the papers still aren't out. So hopefully soon. But it was definitely a worthwhile experience because it allowed us to address questions that we're just able to address now that were not addressable with any other technique. And I think that's really the power of any of these kind of more advanced techniques. Finding a question that can't be answered in another way. If you can answer it using an older method, use the method that's going to give you the cleanest result. If you can only answer it with the optogenetics, go for the optogenetics, but be guided by a good question rather than kind of the flashy technique, pun very much intended.
Okay, so in order to finish, my final statement is worthwhile experience. This exchange of information, protocols, and desires in terms of the future for science. Never science was a singular individual effort. Nowadays, it's even better. You need to get connected. You need to share information. You need to get exchange of people. People going back and forth from labs, from countries, etc. We are highly connected. At some point, you don't need to go somewhere, but sometimes you need to stay there and put hands on. So that's why I think we are here. Not only when you have convergent ideas, but even with the conflicting ideas, sometimes you have, at some point, exponential production, because that's the way science is being made. So just to finish, let me say some applause for this guy. This is beautiful conference and so speakers today. So thank you very much. Mirab, Clayton, and Patrick. Thanks for coming. I already talked to them yesterday when we picked them up at the airport why they were here. They were coming from Rio. Why they not stayed 2 days, additional days in Rio. So I think it's nice to have them after a huge Hebrew meeting in Rio de Janeiro. Tomorrow we'll have a hands-on practical issue. Is basically demonstration or demos on how to play with these protocols. So we'll be at the physiology department, Sala Covian. It's in the first floor near the secretary of the physiology department. I think we can sort of arrange having there like 10 people, not more than that. So if you're interested in coming there at 2 p.m., às 2 da tarde, da Sala Covian, Departamento de Fisiologia. Okay? So we'll be with Clayton showing some of his protocols, some simulations in the computer. We won't go to any laboratories. That Sala Covian is made for that. And Mirab and Patrick have accepted to sort of show some slides of their experience, so we can get together in a sort of informal gathering, okay? So you are all invited to be there tomorrow at 2 p.m. And in the morning we'll come decide with them how to play with time. So thank you very much for staying here till this time. Thanks for beautiful presentations, all of you, and see you tomorrow. Thank you very much.
Thank you.